Hi, thank you very much again for some of you coming for just 2 days. Some of us have been here all week in Addis and kind of feeling the high altitude air. But thank you very much. I mean, we're very, very appreciative, and this is not possible without all of your participation. So, as Alex said, I think we really want to talk a bit about the science, and the science, in a lot of ways, is a barrier to the democratization, the equitable access to initiatives like the Human Cell Atlas. And Alex described just now how we have multiple and profound effects that the scientific community believes will arise from the Human Cell Atlas. And we can see those in regenerative biology, in drug efficacy, and a lot of these things will be able to touch different parts of the globe in very distinct ways. Some of it is in the fundamental understanding of how cellular functions occur, and in others, it'll help to build biomarkers and diagnostic atlases that'll really enable us to have I think, more than a golden era of precision medicine, if that's possible. So we know that there's been really an evolution in technologies that has facilitated the Human Cell Atlas. And this has really been unleashed by the ability to do single-cell RNA sequencing, where we can actually count individual molecules in single cells. But quite importantly, we've also been having a rise in spatial technologies, and spatially resolved omics really refers to our ability to actually visualize in three-dimensional space the positions of the central dogma molecules. And today, what I'm going to try to do for you is cover the advances that the Human Cell Atlas is making in both of these and sort of give a few object lessons on them. I also want to give a plug for some of Alex's work, which I think has done a lot to democratize single-cell RNA sequencing, and to be able to bring it to parts of the world that would otherwise not be able to enjoy it. And that's with SQL. And I can say our lab was really lucky to be able to work with Alex even before this was published, to be able to build these sorts of SQL chips in South Africa, to be able to use them and do single-cell RNA sequencing where it previously was not possible. So the examples that I want to start off with is the Human Gut Atlas. And this initiative covered over 106 individuals, 144 samples, and at single cell resolution, 740,000 cells were sampled. So that essentially was from multiple parts of the GI system. Biopsies, and this, you can imagine what this means in terms of a joint effort between fundamental biologists, clinicians, ethicists, and technologists who work to be able to visualize the data as we've been talking about and analyze this data. So essentially, this was done with a traditional RNA-seq method, which is the drop-seq method, and then single-cell profiles were generated, as well as stains from different parts of the gut to be able to relate what was harvested from gut tissue. And then in situ assays were made to kind of compare the spatial profile to the RNA-seq profile. And so here you can see like the different parts of the gut that were individually sampled. And you can imagine what the kind of basic implications of this are in terms of basic research, but you can also see in terms of types of cancer which affect, or diseases which affect different parts of the gut, as well as different types of inflammatory bowel disease that could truly benefit from this knowledge. So we had the Ramnik Xavier's lab, Alex Shalik's lab, and Aviv Regev's lab really like profiled healthy individuals and used colon mucosa biopsies and generated TSNE maps of these particular cell types originating from those tissues. We also had the Regev and Xavier labs doing basically all cells and neurons that emanate from, so neuronal cells that emanate from the gut. And the ileum and fresh sections of those parts of the lab were generated from, for example, Miriam Murad's lab at Mount Sinai, and imaging was also done at that lab. So this is trying to also demonstrate to you that even though like a lot of this work was done in Boston at the Broad, it also stretched to other parts of the United States where fresh resections were done in the clinic. And again, just bringing this idea that these consortiums that work on different atlases spawn large geographies, and we would like those geographies, especially the gut atlas, to include parts of the world that haven't previously participated in this. And just to really underline that point, there's a specific foundation, the Helmsley Foundation, which released an RFA for a gut atlas, and they only got applications from Europe and the United States. And so this was really unfortunate, but you can imagine that like the utility of such atlases that would emerge from other parts of the world. What did they find from this? Well, they found where disease risk genes act, what type of cells are disrupted, cellular programs that are dramatically changed at individual cellular resolution. They also were able to identify functional modules, communication networks that were disrupted, and this extended to what affects specific drugs that are given, either to treat diseases or generally, like drugs like aspirin, what their effects are, or anti-TNF drugs are on the gut and at the single cell resolution of the gut atlas. So I want to switch now to the lung atlas, because the lung atlas has helped to do something quite unique, and I'm kind of selling the story already, but it's helped us to identify a cell type that was previously unknown. And this cell type is called the ionocyte. And so this effort involved building an atlas, but not only using human tissue, but also using mouse tissue. And you can see here, so the mouse trachea was isolated, but human lungs were also isolated and dissected, and single-cell RNA-seq was performed in them. And you can see that the single-cell RNA-seq yielded this very small population here, where I have my mouse pointer, of a subcellular cell type called the ionocyte. Now, I've expanded the ionocyte group here to show you that it's a very specific gene population, but the interest in the ionocyte is generated by the fact that it is the cell type within the lung that makes the CFTR receptor, which is implicated in cystic fibrosis. So until the lung atlas was completed and until this study was done, it wasn't quite understood that the CFTR gene itself was only expressed in this small subset of cells. So the entire cystic fibrosis disease really emanates from this very tiny population of cells called the ionocyte. The ionocyte expresses FOX1P. I think it's FOXY1 is the name of the gene. FOXY1, like a foxy lady. And so by making a transgene for GFP FOXY1, you can see ionocyte expression, which occurs specifically in discrete portions of the lung. So this enables kind of the construction of what one could call a kind of periodic table of the lung, and definitions of cell types where you can see originally we had HeLoC cells being defined by their phenotypic appearance, and now we have them, this is not in TSNE, but we have them defined as gene expression signatures, as well as their ability to, for example, to perform specific functions like immunomodulation. But again, we must emphasize the discovery of a completely new cell type that emerged from the human cell atlas. So now we shall switch to another type of technology, which is the spatial technologies. And the spatial technologies are really highly important because as much as single-cell RNA-seq helps us to define cell types by their gene expression programs, it doesn't reveal for us the three-dimensional positions, of any of these molecules, nor does it tell us where the cells that are expressing these genes are located. And that turns out to be highly important because we know biology occurs in 3 dimensions. So the technology that drives this is really from the ability, first of all, to be able to take in situ measurements within tissues. And this is from Sten Linnerson's lab in Sweden. And Sten used this barcoding approach where he layered an entire slice of tissue on these pins of DNA, which consisted of this particular architecture of an oligonucleotide. And that allows you to do in situ sequencing right inside this tissue and to be able to reveal the specific locations of where these genes, these key genes, are being expressed within the brain. So it's a little bit of a low bandwidth method, but it's already giving us an idea within this in situ, within the tissue, where these genes are being expressed. Now, we can advance on these technologies more in a more sophisticated way, but what they do require is really different types of combinations of not only single-cell RNA-seq, but different types of imaging regimes and ways to be able to cyclically probe a tissue for multiple genes at one time. And those technologies turn out to be a little bit more challenging because what they necessitate is the ability to kind of do a fish stain in sequence and then remove that particular probe and then encode a new probe into the same tissue sample and repeat this cycle multiple times over. And I think the best demonstration of this type of technology is from Xiaowei Zhang's lab, which is using a technology called MERFISH, which basically encodes a probe on up to 1,000 different RNAs. And then there's a readout probe, which is a fluorescent dye probe, which can then be washed over the tissue and then registered by a microscope and then erased that fluorescent signal and wash a completely new probe onto the same tissue. So the result of these types of experiments are that you can imagine that we can sample thousands of genes at one time at single-cell resolution, but also acquire their single-cell spatial localization within a tissue. Now, of course, there's some technological barriers to that I won't get into at the moment, but you can see how these would be very interesting in general. But then we know that there's high amounts of human diversity, and being able to sample a tissue also means being able to sample human diversity in terms of transcription. And that means that as much as we'd like to detect a gene, we'd also like to detect the variants or variants that we know about in situ by spatial techniques. And sequencing does that implicitly because we are able to acquire the ATCG sequence, but imaging using fluorescent probes has kind of had some challenges in doing that. These technologies that come from Sanjay Tyaghi's lab show how you can actually detect SNP variants across different genes and interrogate different SNP variants, for example, involved in cancers in this cell line, which is really just a demonstration of how the technology would work. But you can see how in this particular cell line, a mutation at a specific point in the EGFR receptor at the level of the RNA can be detected, and even with spatial localization can be seen to be quite different depending on the presence or absence of that mutation. But for me, what I think are the most powerful technologies are technologies that allow us to gain high bandwidth within tissues, because tissues usually have thousands of cells, and the microscopy techniques can only interrogate 1 or 2 cells at a time when they're doing things like MERFISH. So here, unfortunately, because of the lighting, you can't see this very well, but we have an entire tissue here. But by using a certain type of regime in optics called synthetic aperture optics, we're able to capture a very large field of view. And through kind of a physics process called structured illumination, offline, we can zoom in on a particular area in this tissue, and then we can see our probes for RNA fish are able to detect at single cell resolution the presence of specific RNAs. So now these technologies are acquiring a bandwidth that's truly possible to be able to, for us to look at not only single cells with their RNA position at a single cell bandwidth at a time, but actually literally thousands of cells, where this now, after the acquisition of this image, becomes a computational problem where we can zoom in into specific areas like this and actually count individual RNA molecules offline. So spatial technologies are really what will help us to tie the knot between the single-cell RNA-seq data that we get from these different Atlas projects that are ongoing and actually being able to relate that in situ to specific tissues and understanding kind of in a very powerful manner how and where these genes are being expressed and which cell types they're being expressed. And I think it's only actually by looking at the spatial map that we truly understand what cellular state is. And that's something that I think is kind of a debate in our field at the moment, but I think is more and more spatial technologies are going to be used in conjunction with single-cell RNA-seq technologies. So with that, this is just to give you a little overview of the technologies that are available. And I don't know, we're not doing questions, right?
Thanks, Musa, for that really exciting presentation. The science is, I think, well, as far as I can see, it's really exciting. But sorry, Jantine de Vries from UCT, sorry. The question that this generates for me is how, to what extent do you need heavy machines, high-tech labs? So how tech-intensive is this? And can this in any way be compartmentalized? And are there things that people in LMICs without these technologies could also reproduce?
Sure. I mean, these are really good questions, and I guess I threw in that little slide of the sequencing platform that Alex's lab has developed to kind of show you that the cost of this can be reduced quite significantly in terms of at least some of the platforms for it. But I think for people who've been working in H3 Africa who recognize that sequencing and genomics are real barriers in terms of technical infrastructure, those same barriers remain in place for these types of technologies. I think, and I don't know if you want to comment on this at all, Alex, but I think that what we would like to do in projects like the HCA is also use it as an opportunity to be able to attract technical expertise, but also funders to be able to know where to position this type of technical expertise and make it accessible to low and middle income countries.
So, Jantine, it's a fantastic question. The one thing I'd like to add is that some of the technologies are a little heavy in what they require. I mean, we've been involved in developing some that require a lighter footprint. But one of the most exciting things about this is sort of the computational elements of it. So there's now broad accessibility to some of the computational platforms all around the world. And Tim will talk about this a little bit later today. You can use the data that's coming out of these atlases to reinterpret data sets that you already have. And some people are already having, inroads into HCA and using the data that we're generating for their own science without actually profiling anything directly. And a lot of this is now done on the back end of Google Cloud through Terra. Tim will talk about it, but with a Google account, you can actually go on and start to use some of these approaches to analyze your own data, even if you don't have single cell approaches accessible to you at the moment. Sorry.
It's so exciting, Musa, to see how the technology is evolving, and it's evolving so rapidly. And I think this is also an equity issue, because if you have a finite resource, where do you invest your money? And do you do it in a technology that's going to become obsolete very quickly? How do we protect that investment in countries where this represents a big investment locally, but may not be a big investment globally?
Yeah, you've raised a great question. I don't have a parsimonious answer to that question. I can say that, we feel that empowering people at the educational level, at the training level, at the trainee level, allows us at least to build a generation of scientists who are at least adept at using these technologies. I mean, I'm kind of like maybe I'm too optimistic, and I think it's not that I believe in the endless growth model, but I think that poorer countries will become richer and those technologies become accessible. But I think right now we live in an era where poorer countries become richer, but the sort of human capacity base to deploy these technologies is kind of, let's say, a little bit sparse. And we would like the HCA to be an opportunity to change that. In other words, just because you are in an LMIC country doesn't mean that you are not highly skilled in the technology that may not necessarily be freely available in your country.
So I'll just add to that. It's a fantastic question. And I think it's one that we'd actually love to discuss during the various sessions. Things that have come up to our mind are the ideas of creating regional hubs of excellence so that you put a lower burden in trying to figure out where to place those and who would host them. There are some technologies that require a little bit less money up front, but there are pros and cons to everything. And so we want to make people well aware. It doesn't seem like it seems like it would be an ineffective strategy to have samples just simply shipped to a centralized processing facility. We really think that there's a tremendous opportunity here to empower and engage people and enable them to run their own samples. As I'd be happy to talk about at break, one of the biggest problems in running these assays is not understanding the biology you're trying to study and setting up your experiments wrong. So we need to partner with people who are experts in the domain biology that they want to study so that we can actually do these things properly and work with them to use these high resolution technologies to find the signals that are important. Because you get a lot of signals out of this data, as you can imagine. Really what we're looking at now that's just so incredible is we're taking individual cells, we're looking at the expression of every single one of 20,000 genes, and from all of this information using really powerful computational approaches to understand what are the cells, what are the markers that define them, what are the pathways that are active, what are the mutations that are relevant, things that you could have never imagined before. But that means that there's a lot of ability to obfuscate as opposed to illuminate. So we have a couple ideas, but we really want to hear from you what makes the most sense. And I think that, as you'll hear from Tim later, even now, without a lot of the instrumentation, there are easy ways to get involved. And this is one of the greatest things. It used to be that everything was run through some large central servers, but now that everything's been put in the cloud, it really does democratize the computational aspects of it. And so it's a nice way of getting people engaged very early. But to Musa's point, it requires a lot of education so that people understand how to use those approaches and those methods in their own research.
Oh. Katrina?
Hi, Katrina Gold from the Wellcome Trust. Thank you very much, Musa and Alex. I'm really looking forward to today's discussions. One question I have, and I think it came up in London, and I'd be interested for kind of everyone to reflect on this, is to what extent this is about building capacity in single-cell genomics and also spatial technologies, because there has been a revolution in the single-cell genomic world, and the spatial technologies are rapidly evolving as well. And to what extent this is about equity for the human cell atlas and that particular project, and whether actually that distinction even matters, because probably you need one in order to do the other, but it would be good to think about the general points around technology and then perhaps the HCA more specifically.
Yeah, I think that's a really important point. As you say, it came up in London. It's a gray area, as you say. I mean, which is what is equity about here? Is it about equity or is it about technology? I think we need a good dollop of both. And I think it's better we don't opine too much on this, because I don't want to cede our opinions. It's important to get everybody's opinion, because I think every person here from a specific region of the world prioritizes one of those two things differently. I think if you're in a high resource region, you prioritize technology. And if you're in a low resource region, you're prioritizing equity. And so I'd rather leave that discussion open. We have a whole discussion session that's dedicated to that question. Tumbi?
So one of the things that I find confusing is when you're creating the human cell atlas, are you going to be taking into consideration the different disease conditions? I mean, I suspect that it will differ in terms of what is normal versus when you're looking at specific disease conditions.
Intrinsic to equity is the 5 axes that I think Alex described at the beginning are very important, right? But they also include health axes. And we know that disease burdens are different in different regions of the world. And those disease burdens also, to a certain extent, are going to have to be reflected in at least the first draft of the Human Cell Atlas. There will be subsequent drafts where we will get higher resolution of, say, rare diseases that are only found in the Melanesian population. But at the beginning, I think we really want to cast a broad net in terms of the major human diseases.
So Tumbi, let me clarify. So the Human Cell Atlas primarily is associated with healthy samples, but everybody who's actually doing the Human Cell Atlas is always thinking about health relative to disease. And I think one of the most important things is to have an appropriate healthy reference for you to use to contextualize what's changing in a disease condition, right? You want to know what a lymph node looks like from somebody that is uninfected versus infected with HIV for your own research. And so the question then becomes, is a lymph node taken from somebody in Boston an appropriate reference for your own research? I don't know, but it's something that we need to figure out so that we can figure out what are the pathways that you might want to target for trying to eradicate reservoir. But they're things where we need to work with you to understand what are the relevant tissues and questions to ask, so that the atlas really is equitable in what it represents. Let's take one last question, then let's go on. We're going to have a lot of time for discussion at break, but we're going to be talking about this all day. Either way, or sorry, both of you. Go ahead.
Thank you very much. It seems you're going to make a lot of discoveries. For example, you've got in the lung atlas, you've got your ionocyte, which is a new discovery. The question is that, are you attaching patents on these, or can this data be shared across to other people?
So I don't think people are filing any patents on the identification of the ionocyte.
I mean, just all of them, not ionocytes really.
Sure. No, I don't think the discovery of new cell types, I don't think is being used as a way to be able to file a patent. As far as I know, the patent office only does methods or compositions that are not from nature. So if you have a composition of something that's not from nature, then you can get a patent. But if it exists in nature, you cannot get a patent from it. So the Human Cell Atlas, every 37 trillion cells in your body, they exist in nature. You won't get a patent awarded for it. However, if you do find a biomarker that's really important for, say, malaria, and that occurs in a specific cell type of the hematopoietic lineage, well, yes, then the patent office could award you something for that. I think we have our next speaker.
Let's let Christian speak really quickly.
Oh, Christian, sorry, go ahead.
Fine.
Well, I think it's a lot of great work, but I really just want to put it back in the context of, the genomic diversity that you see among the African population, and yet it's great to have those cell atlases. I'm just wondering, in the context of that diversity, how much of those African, cell atlas are you really thinking about putting in? Because that might be, in the end, if you really do not run fast with, African cells, you eventually may have a situation whereby the maps might be eventually biased, and then by the time you kind of start getting African maps, then it represents something a little bit different. So how much do you want to integrate that right from the beginning? Thank you.
So I think that therein lies the reason that we are all here, right? Because I was asked this question 2 weeks ago in Barcelona from somebody watching on YouTube who said, is there a priority for a specific population for the Human Cell Atlas? And I kind of, in a very kind of facetious way, said, well, the priority is North America and Europe. And unfortunately, it's more a question of circumstances than I think of will. And having a program like this in terms of equity is the reason why we can then penetrate these different samples and enlarge the diversity that you're talking about. And that's, for us, I think, is very important, because we need to be able to, as you say, sample African diversity, sample Asian diversity, sample North and Southern American diversity. I think things are extremely important. But the ability to do that in a systematic and organized manner, to encourage scientists to do that, to create mechanisms that enforce those events. That's why we're here. And we hope that today we will actually come together and think of mechanisms that allow us to kind of enforce that in the natural behavior of how the science itself is conducted.
So the one thing I'll add really quickly, and Musa touched on this briefly, one of the things that's so exciting when you think about genetics in the context of the HCA is that really the translation from genotype to phenotype is done through the cell. So understanding cellular identities and using genetic diversity with those cells to understand how these cells are operating on those genetic variants to cause phenotype is something that we're very excited about. And it may turn out that the cell types are a little different in Africa, given, inflammatory environments or, diet or lifestyle. And so we need to understand that, but it's really going to be a way that we can start to use these powerful genetic studies that have been done to understand how you're actually getting phenotypes in humans. So it would be a tremendous loss if we didn't create the appropriate atlases here and layer on the incredibly rich and interesting genetics.